Texas Lutheran University.
All right, good afternoon, everyone. Welcome to the biology seminar. I'm Dr. Gustafson. I'll be introducing today's speaker, who is standing right here. It's Dr. Castro-Arellano from Texas State University up in San Marcos. Dr. Castro-Arellano is a native of Mexico, and he did his bachelor's degree in biology at the National Autonomous University of Mexico. And then he came to the United States for his graduate work to Texas A&M University, and finished a master's and a PhD there in the Wildlife and Fisheries program. And he's worked on a variety of things. We were talking at lunch, and his main focus has been on mammals. So he's a mammologist, but also an ecologist. And now he's really interested in disease ecology, which is looking at the interactions of parasites and hosts and how those might affect disease transmission and things like that. And so he's going to talk a little bit about that at the end. But the first part of his talk is going to be on the niche overlapping bats and rodents. He teaches a couple classes over there, wildlife management.
Yes.
I'm getting this off the
that's
website,
fine. Yep,
so
yep.
Yeah,
that's my class.
Ecology of infectious diseases in wildlife. And he serves on the master's committees of various students, including one who is a recent graduate from TLU, Larissa DiGiacomo, is studying for a master's degree up there right now. And he's one of the directors of her master's degree committee. And he has several funded grants, including a grant to work on how birds that live under bridges affect the water quality of the streams that flow under there. So that's one of his major grants. And looking at various disease organisms as well as that kind of stuff. So please welcome him to TLU.
Thank you. Okay. First of all, I really want to thank the invitation and the nice introduction. What I'm going to try to focus my talk today is mainly a theoretical aspect of my research. And at the end, I'm going to talk about one recent project that's going on. That's still, we're already in our first year. And it's relating precisely that part of disease. And actually, that project relates a lot of my research interests. So the first slide that I open, I always like to put forward what are my interests. And you're going to see just, carnivore ecology. I work with carnivores, conservation biology, meta-communities, which is mainly a theoretical part of my work, and chronoecology. This is the one I'm going to be concentrating today. Landscape ecology and disease ecology. So you first see it, and well, these seem to be separate. Why all these different topics and not just selecting one? Well, the thing is, all these different aspects or all these different topics, they get united through this common axis, which is community ecology. I consider myself a community ecologist, and obviously I do everything in a quantitative framework, and many of the questions that I ask is precisely how the anthropogenic changes we have imposed in ecosystems, how that affect these questions. So, for example, today I'm going to talk about questions about chronoecology, the use of time by a species, and relate that to conservation biology and landscape ecology, and even I'm going to make some connections to disease ecology. So throughout these main axes, these main points, is that it connects all the topics of my research. So let's first get to chronoecology, or the study in time in ecology. We know that throughout the day, the animals go through, go back. Throughout the day, animals go through just like different parts of their environment, even if they stay in a single place. Throughout the day, they go through different times of temperature, illumination, different predators, different access to resources. So it's highly heterogeneous. Even a single environment, even if you're a rodent and you're using a huge expanse of area for your activities, throughout a day you experience very different conditions. And we know that the species have evolved biological clocks precisely that there are selective pressures to guide or have the activity optimized for an area. Let's say that you're a rodent. If you're during the middle of the day, I mean, it's very easy to get predated at this time. So at other points of the night, you might have also the risk from other predators, but it's riskier to be just active in this time of the day. So there are selection pressures to use this section of the day. Now, even though that we know it's important that the discovery of biological clocks has been put forward and one of the major discoveries within biology, the connections to ecology, they have almost no studies. Okay, we know the animals have biological clocks, but we haven't made the connections to the actual ecology of these animals. And within ecology, one central question has been this, the niche overlap. So when you have two species and you have a resource, let's say that it's just like certain type of food, and these curves present how these species use that. So you can have different overlap patterns. Obviously, these species can coexist because, I mean, they use different parts of that resource. Or you can have species that overlap a lot. So that has been a big question in ecology, how the species overlap in the use of certain resources. And one way to answer these questions has been through the use of null models. Those models, what they do is they generate pseudo-communities. So you generate your matrices of a species and the resource that they are using, and through simulations, you generate a distribution. In many of these for many of these instances, you have to use an index that evaluates, that measures this overlap. And then for every pair of a species, you measure that overlap, and then within your matrix, and then you can have this distribution of values generated by the simulations. Now, how does that help? I mean, these old simulations, these are false communities. You're inventing those. Well, if you generate this distribution through your simulations that are within certain bounds, that you know that you're putting restrictions that are biologically reasonable simulations, and then you contrast your empirical value to those, you can actually see if there's less overlap or more overlap that you will expect against this expectation, against this random distribution of value of the index values. So I think it's a very, very solid way to compare your empirical value against these pseudo-communities. Historically, one of the first algorithms that were used to evaluate this, and this is how a matrix will look like. I'm putting in colors, so it's easier to, instead of numbers, I mean, numbers will be just so hard to figure out, to follow. But for example, look at this. These are the list of species, and this is how they use a resource. So even in many of these, they just actually a gradient. What the algorithms, though, just like these ones generated by Lawler from a long time ago, they move the resource states and they repeat these. I mean, nowadays with just like computing abilities, you can generate or do these pseudo communities 1,000 times. Mm-hmm. And then for each one of these communities that you generate, you measure how's the overlap between the species. And then you repeat that again and again and again, and that's how you get your distributions. Well, these algorithms, they are really good for microhabitat and food analysis that they don't have any order to them. When you think about food that just one species uses seeds, another one insects. So doing these randomizations, it's a good biologically reasonable scenario of a simulation. And these are the two indexes that have been used traditionally for this analysis. And there's even a software, a freely available software, if you want to try this, and it's called Ecosystem, that was developed by Nicholas Gotelli, a professor at the University of Vermont, that he is kind of the main person in the area of null models in ecology. So he put that as a freely available software. You can test that perfectly for microhabitat and food analysis. When I was doing my dissertation, I had the idea, okay, I'm going to test microhabitat use and food, and also use of time in the rodent community that I'm studying. And Nicholas Gotelli was visiting Texas A&M at that time. So I went with him, hey, I used your software and got these results. And he just goes, well, you know, there's a big problem. I mean, these results are not good. Why? Just like, I mean, using your algorithm and using your software and following the rules, and you're citing your papers. Well, the problem is that time is an ordered resource. if you think about, for example, let's say that in a group of lizards, and they use different prey items. Just one for beetles, and other ones go for go for worms or different. There's no order in those resources, so you can move those categories for doing the randomizations. It's a sensible option for doing those randomizations, and that's not the case for time, because time there's a high autocorrelation. Animals are very active 1 second, and then they are not inactive the next, and then active again. We don't work that. Animals don't work that way. Normally, activities just like you start growing slowly, have a peak, and then go diminishing. So if you use those algorithms, you're going to destroy that temporal autocorrelation. Also, time has a problem that is circular in nature. So these algorithms are not good. So just like I told Nicolas Gotelli, well, if I cannot use, well, you have to come up with an algorithm. Okay, any ideas? And he told me, no, I don't have any ideas. Just like that. Thank you, that goes one chapter of my dissertation away. So basically what these algorithms, ERIC 1 and ERIC 4 are doing is just you have an activity pattern. They will do this. And this is a biologically unreasonable, just like pattern. So this cannot be used. So that day, I was thinking and thinking, just like, what am I going to do about this thing? I need to either discard that chapter or just like come up with an idea. And an idea came. And actually, I have to thank my wife for that. It's just like she's also a PhD, and we're thinking, and she just suggested this, and just like, yeah, just like, you need a business PhD to suggest to correct the ecologists. So the idea that I came up with is this. Let's see that this is the actual pattern, the empirical pattern. Let's say that what you see in nature, that you have activity peak at the start of the day or the night, the period that you're analyzing, and then it goes down, the activity. Well, what the algorithm does that I came up, it just moves the peak of the distribution one time step. See, it's moving, see, just like checks first this column and this other one. So it's moving one time step each time. And at the same time, because the time is a circular in nature, let's say that this is a whole day. So if you push this once, I mean, something has to happen with this. I mean, you cannot disappear. Well, it just goes to the back. See, all this going to the back. And then later, the original algorithm was only comprised this column. But when I was doing my postdoc, my boss said, just like at the time, I was showing this, I was preparing a manuscript. And he told me, well, you're missing other options, the mirror images. And I said, you're right. I mean, these could be biologically reasonable options. So the algorithm gets your empirical pattern for each species and creates all these options for every species. And then in each simulation, grabs randomly one of these and from all the other species and generates a pseudo community and estimates the overlap and repeats that 1,000 and 10,000 times if you want. It's also a freely available software. I'm going to talk about that in a second. So I termed this, normally in ecology, I don't like when people refer to those to array 3, array 4, which stands for random algorithm tree, and that's not really inventive. So I call it Rosario, just after the Catholic tradition of Rosario beads, that you go in a circular fashion. And just by coincidence, Rosario is the name of my wife and my daughter. Nice move. Yeah, but by now, there's already 15 papers that have used this algorithm besides mine. So it's hopefully we'll keep on going in the literature. So I came up with the algorithm, gave it a name, and now I had to prove that it was a better option. So I did some bench tests comparing to array 3 and array 4. And I found out that, yeah, it's a better option just by its construction, it's a more biologically reasonable algorithm. But also, since it restricts the null universe, it's a more conservative algorithm than array 3 and array 4. So you've got significant results. They are really significant. These two might be prone to give you false answers if you're not careful on how you build up your matrices. Now, I am not a programmer, but when I was writing this paper, and I was using an old version of the program and met a person who it was, in a weekend, was able to make a small program that we call it time overlap. And this program uses both the Pianka and Sekanovsky indices, and we published that just as a paper, just the methods. I mean, that was an unexpected chapter of my dissertation, that actually the method was just published as a paper by itself. And as I said, the software is freely available. You can download it from my website, both at Texas State and the place where I was doing my postdoc. The links are also in there in the paper, and I think it's one of my favorite papers. It took a lot of time to develop these ideas. So, okay, well, that's very nice in theoretical. I mean, I always tell that's very nice to see these things in a theoretical realm. But it's more, I think people get a better idea of what the algorithm can do and the potential applications if you use examples. So I'm going to put those two examples, one first with a rodent assemblage, and then another one with bats. The first example is coming out of my dissertation work, and the other one is posterior work when I was doing my postdoc at the University of Connecticut. For my dissertation, I had got my data from El Cielo Biosphere Reserve, which is our reserve that it's very unique in many aspects. It's located right in the transitional zone between the Nearctic and the Neotropics. It's in northeastern Mexico. It's about 300 miles from the border. It's a pity right now that we cannot go at this moment, but I mean, that area of Mexico is not really safe right now, but I don't hope that that's not going to last forever. So after things calm down, I strongly suggest you go to this area. That part of Tamaulipas, of the state of Tamaulipas, most of Tamaulipas is just like Texas. It's just a big coastal plain. The big difference is that there's this chain of mountains that is in this part of the western part of Tamaulipas. And while you're driving to those mountains, I mean, I was coming from College Station, and when you get close to this mountain, it's almost like a wall. You're driving to a wall. The mountain goes from 200, the reserve starts around 200 to 2,000 meters above sea level in a very short distance. You can actually hike that in a day. And that's pretty, I mean, it's a big, really strong hike. I don't think I can do it now. When I was in grad school, I was able to do it several times, but not anymore. And you go to different vegetation types. You start with the coastal plain. I mean, will be just like here in Texas. And then you get the coastal plain scrub. Then you get tropical subdeciduous forest in the first part. And then cloud forest, because the way these mountains are oriented, all the moisture from the Gulf comes, and then it just drops the water in there. And you create these really nice cloud forests, which, by the way, is the northernmost neotropical cloud forest, but also has combination of Nearctic elements. So you create a really unique place within the reserve, which is, I mean, it's really large. It's 144,000 hectares, which is large, but it's not the largest reserve from Mexico, or not even, I mean, it's small compared to Yellowstone, for example. But within the reserve, there are jaguars, mountain lions, and black bears. So you can see the combination of animals that coming from the south, from the Neotropics, and also from the Nearctics. So it's a really unique in that regard. And also, you get, I mean, after the water, all the water drops in here, and the clouds keep on going, then you have a dry environment on the other side of the reserve. And the last part of the Chihuahuan Desert arrives at that reserve. So it's really that combination of environments is pretty unique. It's a pity that just cannot go back. I would love to continue my research in there. So when I was doing my dissertation, I selected two sites. One was just right in the border in tropical subdeciduous forest, and another one in cloud forest. And the idea was to test. I wanted to see how the two rodent assemblages compare in their use of time, because I knew before starting that they were really contrasting assemblages. So just a couple of photos of the place. See the tropical subdeciduous forest. It has a lot of tropical trees. We actually had to open these trails to put the traps. So very densely vegetation, just like many species from the tropics, very moist, humid. And then you have the cloud forest. I mean, a very completely different amount. And the distance between those two sites is 5 kilometers, straight line. It's nothing. It's nothing. and see the other one was 200 meters elevation, and this one just like not much higher, but very contrasting vegetation, just like in each one of these sites. So basically what mammalogists do, it's what we do, we get our traps, we put them in the afternoon, we put the bait, and then we go back to the camp to have a beer and just have a fun night, just like go sip some whiskey, and then we just check them in the morning. So the night is almost like a black box. We don't know what's happening. So for my work, I really wanted to see what was the activity for each species. So I came out with this protocol, which I call interval trapping, which is checking traps every 2 hours. Now, I'm going to tell you, I broke the spirits of more than one assistant. One of them that I had from A&M, just like after 1 month of being there in the field with me, one day he said, I don't want to do this anymore. I'm getting out of here. Just get me to the bus station. I put him on the bus, and he went back to Texas. He came back here. He decided that it just was not, he didn't like it, just like it. And it's hard, I mean, because basically what you're doing is this. So you're checking traps every 2 hours. The problem is not staying awake. The problem is staying awake between the last time periods. We invented I don't know how many things to try to keep ourselves awake, just like at these last periods. And when finally we did the last one, we just hit the bed and try to sleep. The problem is that we're staying at a biological station that was just a roof, and during the day it will get so hot that it will get you out of bed. So that was the challenge. So what happened, just like after all those checks, or when you were doing this checking of the traps, if you get a successful one while doing the checking of the traps, what we did is we took the trap out and we replaced it with a fresh one. Why? Other people that have studied this, they have done the easy way. They put timers in the traps that they detect at the moment the trap closes. The problem that I had with that, and actually spoke with the person that published a paper for that, let's say that you put 100 traps here at the start, and you get lots of rodents. Let's say you get 30, which is a lot of animals in a night. The last period, you're not sampling with 100 traps. You're sampling with 70. So how you're going to correct for that? So there's no other way but to stay awake and exchange your traps. So it was hard, but I think it paid off. So I did almost 7,000 trap nights, which translates to almost 41,000 trap periods. So, and you can see it's just like, it's a lot of work. We got almost 500 captures. The success rate is about what you will get into these environments. The traps were left open during the day to see if there was any species having activity during the day, but we had a single event of diurnal activity, and we detected a total of 12 rodent species. Now, as you can see, the cloud forest has less species than the tropical zoos. I mean, this is typical. Environments that are more nearctic, they tend to have less species than these ones, that they're more tropical, that have more diverse species. Now, not all of I was not able to get good data for them. Many of these species are rare. So I will only get 1 or 2 captures for those. Obviously, you have to discard those because, there's no statistical power in comparing those. However, I was able to get data for several species. Now, in the cloud forest, I had two dominant species, which were Peromyscus, this is Levipes, and another one, an endemic, which is Ocraventer. And for those, as you can see from this is the number of percentage of captures and the time periods. And with the statistical tests, I proved that they have almost the same or not different activity periods. So it seems that the cloud forest, they can share the night. These guys, they don't separate the night. However, it was very different for the tropical subdeciduous forest. There were some species which will start late in the night, other ones that will start early and then will fade down. Another group of species will be crepuscular in their activities. See, they will use the start and just like early in the first hours of the day. The same for this one. And another one will use the center. So if you look at just by looking at those charts, do you think, do they overlap or do they separate? And just by looking at those. it's hard to judge. I mean, it's just, I mean, some people will say, no, I think they separate. No, well, I think they overlap. That's the whole point of the algorithm. So now, with this data, with this empirical data, this is the distribution created by the algorithm that I came up myself. And this is the overlap detected, or I mean, this is the overlap value for the empirical data. And as you can see from it's significantly smaller than the simulations. What that means is that the rodents divide the night. And this very, I mean, with around this very times of doing different combinations, it's very, very powerful. So this is the first time that someone proves that species show non-random temporal partitioning. So basically, they divide the night. No one has shown this before because mammalogists, as I say, normally we leave the traps and we forget about those, and it's not until the next day that we check them. Now, since I was doing other work like microhabitat work and just like the stomach contact work, I contrast the results to the microhabitat. So let's say at this the community, or this assemblage where rodents divide the night, also I did some microhabitat analysis, and I found that the animals divide in 3 different groups. Peromyscus pectoralis, it's a species that inhabits deep inside the forest. Leiomys, Oligorhizomis, and Oryzomys cowesi, these ones are ecotonal species. They like the borders between the forest used by Peromyscus pectoralis and the open habitats used by Biomys and Sigmodon. So they also divide my microhabitat. If you pull the activity patterns of species at the forest versus the ones in the open, they have completely opposite patterns. So that speaks about the limitations that some species might have for each one of those environments. And it even gets more interesting, well, just to compare for the species that are in the cloud forest, I found that they don't separate by microhabitat. They use basically the same microhabitat. They use the same hours of the night. And I also found that they use the same food. Their data is not here, but they use the same food. I'm still puzzled to see what divides these species, although that assemblage has much less species and is heavily dominated by Peromyscus levipes and Peromyscus ocraventer. Now, at this time, I'm not presenting just the results that I have for that other part of the research. But while I was doing this, I have a friend that was doing his PhD on hantaviruses in the University of New Mexico. And he asked me, hey, are you sampling rodents? Yeah, I'm killing a lot of them. So why don't you just collect this blood from them and we can test if they have hantaviruses? Yeah, sure. So he sent me the materials. We get the blood samples. We send them back. And I have found a huge 23% of the Peromyscus levipes we capture at the cloud forest were infected with a hantavirus. So that has implicated, now that I'm doing disease ecology, doing this in retrospective, I mean, it wasn't the purpose of the dissertation, but we had another project later and we're still analyzing the data. Obviously, in an environment in which you have a species that has a hantavirus and it's sharing the same microhabitat and the same hours of the night, we expect, when we return to do a later project, we expected that, yeah, they're going to find also more of the other species infected. And yes, we found more of the other species. We're still working to publish that. Whereas in the tropical subdeciduous forest, we haven't found a single species with hantaviruses. But those divide themselves. They don't encounter. So the chances for a hantavirus to jump among these species is very, very slim. So now that my research is doing mostly disease ecology, I see that the background and the work on just theoretical ecology is becoming very, very handy. Another thing to compare within this assemblage is I have the cloud forest in here and the tropical subdeciduous. Even another thing that puzzles me in the cloud forest, these 2 species that are the dominant ones, they are very, very close in their body mass ratio, so they are very similar in size. Whereas in the tropical subdeciduous, see, there's a big gradient. Both the smallest and the largest of the species are the ones that were using the crepuscular parts of the, see? They have big separation in body sizes, although they use the same time of the night. And the ones that had the highest separation in the night are the ones that have almost completely opposite activity patterns are these two that are highly similar in body size. So it seems like the structure in this assemblage is not at random. It's really a lot of structure behind this rodent assemblage at this reserve. And well, what does that mean for, as I was mentioning, for disease ecology? Now we want to compare this, the next step. As soon as I am able to return to Mexico, I want to put telemetry colors and start chasing these species to see, to generate these, what is called contact grids in disease ecology. Compare these two, in this case, ideas, there's two environments through different months. Two environments in which there's a lot of contacts between individuals, and it could be different species, could be the same species, and compared to that. I think that what I'm going to find in the tropical subdeciduous is something like this, and in the cloud forest is something like this. This has obvious implications for this dissemination and maintenance of virus that can jump to humans. So that's going to be the next step as soon as I can return back to Mexico. And actually, we're going to submit, hopefully next year, a proposal to NSF to test for this same idea here in Texas with another different system, comparing an area that has been highly affected by agriculture, it's highly fragmented, and an area that's more pristine. And I think where I'm going to head this is just like in the area that's fragmented by humans, just we force the animals to just live in smaller patches, and we're going to have higher contacts within them against a grassland that is just more open, and we're going to find something like this. That's our hypothesis. So we'll see in probably 4 or 5 years now if we get the money. So another example. So let's talk about just bats, now changing from rodents to bats. And for this one, it's data coming from the northern part of Peru, from Iquitos. And that one, we're using the most abundant phylostomids. There are frugivorous bats. I mean, bats that only eat fruit. And we're comparing 3 different habitats. And we use 7 time intervals to sample through the night. And instead of traps in here, it was through mist net sampling. Basically, in Peru, in Iquitos, we found this archetypical situation from the neotropics in which people open up areas for agriculture. And they use them for a while, and they are abandoned. After their soil is not good anymore, they abandon those. And then secondary vegetation takes over, which is purma. And then eventually it comes back to forest. And you can see those 3 in in very close proximity to each other. So in here, after this, we did all the sampling for these bats from the 3 different environments. We restrict our analysis to the most abundant species because obviously they're the ones we can get good data for. And in this chart, the first bar is one species which is going to be the same one in the next step, is the time period. So you can see the activity pattern of this species will be this bar, then this one, then this one, and then keeps on going out throughout the 7 time periods. What is relevant in here is see how do they group. This is contrary to the rodents. This is completely the opposite pattern. The animals seem to have just like pulling all their activities at the start of the night in the pristine environment, which is the forest. But look what happens in the secondary vegetation. It starts to get, it's not the overlap. It starts to get more lax. The activities are more distributed throughout the night. And the open environment, the chakra, it's a completely different pattern of overlap. And it seems like the peak of the activities is just like later in the night. So applying the algorithms, again, the same situation. Just by looking at the charts, it's very difficult to see if they are overlapping or not, or in a statistical sense, just like to be sure. Well, when you apply the algorithm, we found that in the open environments, the ones that we affected as humans, depending on the season, you have instances in which you find random overlap. But if you compare to the forest, independent of the season, or if you combine them, you always get chronocoincident activities. So they always have their activities at the same time. So obviously, here time is not an axis for separation. Animals are not dividing the night. In here is the contrary. It seems like the bats have common constraints. They all have the same problem, and they need to time their activities at certain times of the night. Our hypothesis was that when we published this paper is that since they are frugivores and fruit is a very poor, nutrient poor and energy poor food, they need to get lots of it. So they have spent most of the night in a cave or in a roost, and they need to go as soon as possible, as soon as the night falls, to go and start eating, because they might have some energetic constraints. So that's the question, do they really need to start early in the night? And what happens if, as humans, if we modify those environments and we don't allow them to start their activities early in the night? And the reason we believe that in the open environments they start later is because the chance to get predated, or the chance of predators getting them, is higher in open environments. So by opening the environment, we force them to start late. Now, it's a good question. This is still an open-end question. Does there really, is putting a constraining fruit for frugivores? I did this research when I was doing my postdoc, and my former boss liked it so much, the question, that right now there's a student in Costa Rica, one of his PhD students, trying to answer that question, obviously doing this in an energetic framework. So we'll see in a couple of years what happens. So that's from the data from Peru. Later, when I was just here in Texas State, I got contacted by a friend from Cuba, and he saw the papers for the previous papers and say, hey, I have data for this. Can we analyze that? Sure, let's try to analyze it. We'll see. And I was thinking, we're going to find the same pattern as we see just like in Iquitos. Well, in here it's just five Phytophagous bat species, 5 frugivorous. There are nectarivore and frugivore species in this group. We are comparing highland and lowland. Highlands are the pristine environments, and the lowland are more human-affected environments. We have wet and dry season and 8-time intervals. So the data is very comparable to the data from Peru. So we can really make comparisons across these two data sets. And it comes from a reserve here in northern Cuba. Well, there goes our hypothesis. It seems like even independent, the black bars is the highland, the pristine environment, and the white bars is the lowland. Seems like in Cuba, they don't care. They don't need to be early in the night. See, the peaks are in the middle of the night, the activities for those. So what's happening? I mean, well, this is an insular system. How the resources are distributed. These animals roost more in caves than in the other, in Iquitos, most of them, they roost in trees. So that's another question. What's the distance from the place where I'm roosting to the food source? So that might be another way that we might be affecting the environment of the bats, that making them forced to travel longer distances in open habitats as compared to just when you have the pristine forest. So again, this paper opened more questions than the one it solved. And right now we have, and also we use the Rosario algorithm, and it was consistently, even independent of all the combinations that we tried to do, removing some species, because Monophilus redmani also uses insects. So we're thinking, we might be putting some noise if we put this species in. Well, so we remove it. We test both habitats with different seasons, and see, even with all the combinations that we did, it was highly consistent that they overlap more than you will expect against a random expectation. So they overlap, independent of habitat, dependent on season, and they use the middle of the night. So what's going on? So I think the answer is going to come after we compare many different systems. And in the website that we have our program, just like we put an open invitation for researchers, because many of them, they might already have this data. Many bat ecologists, they go and they put these nets, and they actually register the time that they capture. So we have been in contact with tons of different, we have right now close to 27 different studies joining, and hopefully next summer, together with the other postdoc that was at UConn at the time I was also there, we're going to try to assemble a major paper trying to compare and see just like what's the dominant pattern, because we know the details of each one of those pieces of work. And the outer line in that one is going to be interesting. It's just going to be just so many authors in there in that paper. So as you can see, the algorithm has a wide application. Obviously, it's not restricted to the rodents or bats like these examples that I put. Other people that have used it, it's always a nice surprise when you see other people seeing your algorithm. They have used for pollinators, just like how pollinators overlap of just like of our resource, different insects. They have also used it for butterflies. And I think I got into contact with a person I wanted to use for hummingbirds, but I haven't seen, I don't think this person has published that research. Also, it's not restricted to any given temporal cycle. It can just be, the examples I'm putting are circadian examples, but you can use also a season. I know it's just depending on how you build your matrices. And also, I got contacted by a person doing studying the fossil data, and say, hey, do you think we can also use this for abundance of species through longer time periods? And that led me to think, like, yes, I don't think there's any limitation. And so this person is going to try to see if there's change in the abundances of foraminifera species over longer periods of time, and it's going to relate it to Milankovic oscillations. I really look forward to seeing that paper. And another person asked me, hey, what do you think about the data? And they put me the question, hey, what about fish abundances along a river? Well, the only problem I have with that, you really have to see if your spatial data is really highly autocorrelated, and if the circular assumption of the algorithm, if it holds. If you think those are biologically reasonable assumptions for your model, I think the algorithm is better than array 3 and array 4 that will destroy any spatial autocorrelation in the fish abundance in a river. So that one is still pending. I don't think he's pursuing that, but I think that will be another avenue. The interesting part is probably data is already available. And actually one of the big things that I like to do, when I get alerts of new papers that have been published, they are always looking to see if people put the matrices of their activity patterns and say, oh, it's almost like free data. And I'm reviewing right now a paper of a group of researchers from India in which they, with camera traps, they have the activity patterns for carnivores in that reserve. And they have carnivores and also herbivores. They have at least 15 species. They are testing their activity. They want to see if there's any relationship with body mass and also the type of food that they're using towards their activity. But they don't put the data, but I'm sure they have that data. So as soon as that, I think the paper is just going to get out. I mean, it's well written, the data is solid. So as soon as it's out, I'm going to contact them. Hey, do you think you might want to share your data and put out together? Because also a PhD student from Mexico contacted me early in the year. Hey, I saw your paper. And do you mind reviewing my publication? Well, she had good data, but it was completely wrongly applied. She couldn't understand. She didn't understand at that time exactly how to apply the algorithm. So I said, well, you have to completely redo your analysis because of this, and that. So they told me, hey, would you like to help me? Yes, certainly. So I redid all the analysis, and they are studying a reserve in central Mexico in which they have 6 carnivore species of body sizes from very small cats to all the way to jaguars. And we applied the algorithms, and we came up with some interesting things, and we're probably close to submitting that manuscript. So it has really given me a good sense of working in academia in a very collaborative way. Like, for example, this paper from Cuba. I know this person, I have met him, but all the work I have been on, we have been exchanged just via email. So it has been really nice being in contact with people that, I mean, they are the experts at the systems. I'm just helping them, just like focus on a specific question with the algorithm that I came up with. Again, just like same idea. I'm collaborating also with people studying rotifers. I have already analyzed, I haven't had the time to just build the manuscript. Some researchers from French Guiana, they have 25 years of data they put these traps, and they had bills, and they check them every 2 hours during the day. So if they have thousands and thousands of captures. So they actually put the raw data in their paper. So I contact them and say, hey, I think we can analyze this. So I have already analyzed that, and just need some time to publish that. And again, just like the example I was putting with bats, now I did at that time that I did the original ESA slide, it was 4 different. Now it's 27,000 people that have contacted us to join this major effort. So that's going to be really, really interesting. And also I'm on the co-chair of a dissertation that's working on the butterflies from El Cielo Reserve. So that's the part that I want to talk about the theoretical part of my research, which you can see that it also has connections to to landscape ecology, conservation biology, and even to disease ecology. And that's one of the big goals. I really want to go. No one in the disease ecology realm is just like applying all these ideas from the activity patterns of animals towards disease. And it's a huge part. The contact rates between animals, between individuals, is a crucial element of how a disease is going to spread between the individuals. And that area, even if it's abandoned in ecology, it's almost non-existent in disease ecology. So I hope I can start filling that gap. So I'm going to talk about another project that might be more attractive than all this theoretical mumbo-jumbo that some people think is boring. I think it's fascinating. And it's about the project that I have right now with they're doing in Puerto Rico. Basically, the question is that we're trying to figure out if an invasive species has, and this is the culprit, this animal, if it has a role to sustain the cattle fever tick populations in Puerto Rico. Now, I don't know if you're aware of that, just like this tick was here present in the United States, and it was eradicated a long time ago through a series of just quarantine methods and just control. The tick is not a problem by itself. The problem is that the tick in its gut has a parasite that when it passes to cattle, it kills it. So obviously, if you're a cattle rancher, it's just bad if you get ticks that start killing your cattle. And that's what's happening in Puerto Rico. They are losing lots of cattle head because of this disease. And I suspect, so they suspect that that's the disease. Babesia is called, babesiosis is the name of this disease. They suspect that they have that problem. And I think they have more than one problem. So we're going to test for several diseases. And the idea is to see if this animal sustains in the environment. Now, Puerto Rico is a very unique case because there are no native land mammals. The only native mammals are bats. And bats don't overlap with just farms and just cattle ranches. So what other animal could be sustaining these ticks? Because what they are doing in Puerto Rico, you have a pasture in which you have your cattle. So you get your cattle out, just wash them with these just like insecticides, acaricides, and remove the ticks. And then you quarantine that pasture. You know that there are ticks in there. But if you don't put food in their cows, they're going to die because there's no food after some months. Well, they did that thinking that that was going to solve it. They put them back and they get ticks again. So what's going on? Well, probably this animal is keeping the ticks in the environment. That's what we're trying to prove. This animal, the small Asian mongoose, was introduced in Puerto Rico in the 1800s to control rats in the sugarcane fields. At those times, you know, everyone knows about the parrots, they like rum. Well, all the rum was being produced was here, and just like in the Caribbean. So they had huge expanses of sugarcane fields. That was the main product of just many of these islands. Cuba, just like Haiti, Dominican Republic, all those islands, they were producing rum. And they had a lot of rats. So someone had a really bright idea of bringing mongooses to control them. What's the problem with that approach? Well, it's independent of this invasive species, the rats are nocturnal. These are diurnal. So they don't meet. Or they meet at the crepuscular hour. So they do kill rats. I mean, they can kill rats. And they actually, there's some proof that they actually reduce somewhat the levels in the sugarcane field. However, they don't kill enough rats to be a biological control. So they didn't serve the purpose that they were introduced for. And they decided that the local endangered coquilles was a much easier meal than going for a rat. So now it's a huge ecological problem with this species because it's killing native animals. They are everywhere, and also they are the prime reservoir for rabies in Puerto Rico. There's more cases of rabies in humans from these bites from this animal than from domestic dogs over there. So that's that big, I mean, it's a big, big problem. And I think they are also part of the problem to spread a disease that they have in the kidneys, leptospira. It also affects the cattle. So we got a big chunk of cash. We got $1 million from the government of Puerto Rico that went directly to the USDA lab that I collaborate with, that is just like here in Texas. And they are doing the work on their cattle, and I'm concentrating my work and trying to figure out if this animal has that supposed role. As most of you can think, I mean, it sounds really nice, Puerto Rico, the Caribbean, and But actually, the places that we work are less than being pristine, and many of these places are full of cow pies. So, but we have to work in those places. It's nice working in the environment. Just you have to be careful where you step in these farms. And you see the degradation, see the forest, and see the degradation that's going on. The mongooses, they love this interface. They are in here, they move in here. So what we're doing right now is putting telemetry. We're doing lots of trapping. We actually, these are photos from this summer. That's my grad student she's leading this project. She spent all the summer in Puerto Rico with an undergraduate assistant, paid also by the USDA. That was really nice that we got that support. And we put transects of 50 tomahawk traps. We get the animals, and for every capture, we obviously would register on the GPS, and then we put on a collar, a radiotelemetry collar. And then after I was there at the start of the summer, we trap off the animals, we put the collars, and then the students left behind and did the rest of the summer, they did this. They just keep following the animals. The whole idea of doing this is we want to see, first, we also search the animals for ticks. We trapped them again at the end of the summer, and we searched for, and we did found ticks in them. So that was, when we found the first ticks, I actually took a photo of my cell phone and sent it to the person that I have to respond to, the head of the laboratory that I work with. And it's just, we have to open the bottle of champagne. No one has proved this connection. And obviously they are giving us just like a lot of money to try to solve this. And it seems that our hypothesis is correct. Now we have to really see, finding them, the ticks in the animals is just part of the deal. We have to see now, how do they move and overlap with cattle pastures? And I think they overlap a lot. I think just by looking at the first pass of the data. So another question, I have another grad student that the animals that we collect the kidneys, and she's doing genetic work right now to look for leptospira. Leptospira is a bacteria that is in the kidneys of many mammal reservoirs, and that's another disease that can jump to humans, or can jump to the companion animals, or also to cattle. It causes abortion, or also just like, it affects cattle so much that you don't get as much milk or as much meat. So I think the Puerto Ricans called us for one problem, and I think they have more than one over there. That's my suspicion. We still don't have any results. We're still starting to do this search. And I have another student that's looking at the endoparasites. It's going to open all the stomachs, all the digestive tracts of the animals that we collect to see if we found any parasites that can jump also to humans, that can jump to cattle, that can jump to, they are shared with dogs. And finally, another student is looking at the food habits. We're going to see if we can get money to see if we want to identify if they are eating endangered cookies by genetic methods. So we'll see if we get the money for that. So I just want to thank I need to mention all my funding sources, the government of Puerto Rico, the USDA, Texas Parks and Wildlife for giving access to some work that we have been doing for other purposes. I have other research here in Texas, and also the research that we have that was mentioned at the start, and all my collaborators. That anything so far that we have done, wouldn't have been possible if it was for all this group of students that are really hard workers. So I think that should be it.
Okay, we have a couple minutes for questions.
Yes.
We were talking earlier about Ebola and training. Would you comment about what's going on with Ebola and how that got into the...
Yeah, I actually, this week I was talking with my wildlife management class, and I wanted to really emphasize the point that this situation we have right now with Ebola and that most people are getting really worried about. First, I wouldn't worry about it. The case in the US is going to get contained. We have really amazing expertise from the CDC. The problem is going to be the people in Africa. They have that outbreak is really scary for those people living over there, because obviously they don't have the equipment, the facilities, the training to control. And where this virus came from, this is just a problem of mismanagement, destruction of habitat. And just people tell me, what's the connection to conservation biology? This should be an issue for medicine. No, it's not an issue. It's an issue of conservation biology. These viruses occur in what we suspect there are 3 bats. I mean, we still are not sure, but there are 3 prime suspects, which are 3 bats, 3 fruit bats, actually really large ones that occur in Africa. And in those bats, the virus exists in a natural way. The bats have immune systems that can deal with this virus. It doesn't kill the bats, but the virus can jump to chimpanzees. It can also kill. There has been outbreaks of this virus in gorillas, and after it jumps to other species, when humans go and they try to trap chimpanzees or gorillas or any of those species, also it can jump to some antelopes. So if people go and hunt these antelopes and that antelope was infected, and you're just field dressing that animal and you caught, there you got. And then you get it to the next person gets to the other, and then it spreads like wildfire. So it's destruction of habitat. We're getting, as humans, in places we weren't before. We're using wildlife in a non-sustainable way. People are trafficking wildlife. Why this outbreak is coming in West Africa? The virus is originated from Central Africa because the illegal movement of animals from Central to West Africa. So it's an issue of habitat destruction and mismanagement of habitat or environment. So, and we're going to have more on those. That virus is in the news, but I can give you probably 50 other things that are just there that can jump like this virus. So, any other question? Yes?
Do you think there's applications for like microenvironments, like gut bacteria, like with the algorithm?
Yes, it could be. I mean, it's not restricted to any taxa. And in bacteria, you don't talk about activity, but you can talk about growth. I mean, just how growth, just like the colonies, just how much you can take almost like a snapshots and how much space they use through different time periods. Obviously, you have to reduce the time frame, the time intervals that you're analyzing. For this algorithm, it's important to see what species are you analyzing. You're getting into the matrix, the time intervals, how you're cutting, and then the time extent. If you're not careful how you build those, then you come just with trash. I mean, with any model in ecology, trash goes in, trash goes out. So you have to be careful what you put in the model. So because the model is the software is gonna give you an answer. Yes, the program is interpreting and just putting in the right context that answer. Mm-hmm. Any other? Yes.
You kind of mentioned a lot about populations from kind of like Central America. What about, is there any data on like U.S. bat colonies, like the one in Austin? Have they had data collected on those as well? Or?
Oh, for activity?
Yes.
For bats? The only work this major work that we're getting, just like different studies together to analyze, the only one that we have got so far is just from a group of bats in Arizona. The thing is that the bat communities or bat assemblages here in the US and the Arctics, they are not as diverse as those. I mean, they have been heavily studied, but if you have only one or two species, then the algorithm, the idea of it is to compare the overlap in species-rich assemblages. So it's not a question. The algorithm doesn't apply to this. It's mostly towards species-rich assemblages. So there are lots of data for bats here in the US, but it's a very different question. Mm-hmm. Yes, Chief.
Quickly about the graduate program at Texas State.
Oh, yes.
What would a student have to do to get in? I see one of our students up there on your
really?
list.
Oh, Larissa? Yeah,
Yes.
Larissa participates. She's participating in the Texas DOT funded project. Well, the first thing I will say, if you want to get, I mean, you see, if you have interest in grad school in Texas State, go to the website, check what's the research interest of each one of the faculty that is there. Because now, one of the big requirements to get into grad school over there is that you need the letter from the advisor to the intent to be your mentor. If you don't have that, if you don't have a person that wants to be your mentor, that application is going to get discarded. I mean, it's just like, because in the past, some people were accepting the program just kind of as orphans because no advisor. And then it takes time for them to get them, and a master's is very short. So I strongly suggest go and see what people are doing, what research and if there's something that really has interest. I mean, you see that there's something that you can share, that interest, go and talk with that person. I mean, I think all the faculty, that's one of the reasons I like that place. All the faculty are really good, they're really approachable, they're really nice persons. So just go and talk with them and see what are your interests and see if they have projects. I mean, for example, I can always take students. I'm limited in the projects that have funding that can go to nice places like Puerto Rico, but I have tons of projects on theoretical data that could just go to the literature. And I think there's an underappreciation of the students for those kind of papers, and I think you can publish higher impact papers without data in the theoretical realm instead of just having the portrait. And we're always, this summer, we needed so much help other than Puerto Rico that I actually have to pull the students from other projects and send them over to Puerto Rico. So there's always opportunities to have experience in field work. So that will be my suggestion. Go and check the website of Texas State.
All right, thank you.
Thank you.
For more information, please visit tlu.edu.